Dear student, I am Anil Srivastava from Government VYTPG Autonomous College, Durg, Chhattisgarh. Today we are going to consider the topic insect wing venation. This topic is beneficial for the students of MSc Zoology and especially for Entomology paper. We all know that insects having some wings, and on the basis of presence of wings or absence of wings, we can divide insects into two categories: Atericota, insects without wings, and Terracotta, insects with wings. Generally, in the body of the insects, two pairs of wings are found at the dorsal side of the thorax, derived from the integument, which is supported by hollow veins, and the wings remain articulated with the thorax. Inside the wings, there is a distribution system of the trachea, which is known as the veins. And trachea is nothing besides the respiratory systems of the insect responsible for transfer of oxygen from atmosphere to different part of the tissue and to transfer the carbon dioxide from tissue to the atmospheres. The distribution of these wings as a vein in the body of the insect, in the wing of the insect, is very typical. And first time it was characterized by two important entomologists, J.M. Comstock and J.G. Needham. The distribution pattern of this vein in the insect is variable, but the basic concept is almost the same among all categories of the insecta, among all orders of the insecta. Here, a complete system of vein of a wing is called venation. The recognition of certain features of the venation of the wings of insect, which occur in the more generalized form of a large proportion of the order of class Insecta, has enabled Comstock and Needham to present a hypothetical type to which the wings of all order may be referred. It represents the supposed arrangement of the trachea in a wing of nymph of the primitive winged insects and thereafter among all adults also. Here, it will be seen at a glance that hypothetical type differ from the great majority of living insects in the possessions of a larger number of wing veins than in characteristics of them. It also differ, and in more striking degree, from most of the insects of order Neuroptera in having a much smaller number of wing veins than in exposed by them. So first of all, we should understand that what is hypothetical system of venations proposed by the Comstock and Needham in the Insecta. Here, as the Comstock and Needham proposed, in the body of the wing of insects, 5 or 6 types of major veins are formed. The first one is the costa, represented by letter C. The costa normally considered as the first vein, usually marginal, but sometimes it may be submarginal, and this is unbranched vein. The veins may be convex and the concave, but the first one, the costa, is the convex and remain associated with the humeral plate of the wings. The second is the subcosta as proposed by the Comstock and Needham. Generally, it is represented by the SC. This is the second vein, normally unbranched, but sometimes in some cases of the insect it may be branched, but it is normally unbranched. And the basic structure of this subcosta is concave and found associated with the distal end of the neck of first axillary. The third is the radius. This radius vein is the strongest and divided into 5 branches. And the main stem divided into first R1, this is the convex branch which pass up to the wing margin, and second concave radial sector represented as RS. Radial sector further divided into 4 veins: R2, R3, R4 and R5. Here the radius remains associated with the anti-radiant of the second axillary. The next one is the media, represented as M. This is considered as the fourth vein of the insect wing, divided into convex anterior media, MA, which further divided into two branches, MA1 and MA2. This total structure is the convex organization. But the posterior media is concave, and which further divides into MP1, MP2, MP3, MP4. So MA meaning for the anterior media and MP for the posterior media. And complete media remains associated with the median plate. The fifth one is the subcutis. Cubitus, not subcutis. Fifth one is the cubitus. And it divides into two branches. First, convex subcubitus, CU1, which further divide into two branches, the CU1A and CU1B, you may represent it. The second concave branch, unbranched, is the cubitus, referred as CU2, and it is found associated with the distal median plate. And the last one is the anal vein. The Comstock and Needham have represented three anal veins, A1, A2 and A3, which are usually convex. But in some cases A2 may be concave, not in all. But there is some controversy. Later the Snodgrass advocated that the first vein, meaning 1A, does not belong to the same series of the anal vein, but it should be designated separately as a postcubitus. So this is the basic plan of the venations given by the Comstock. This figure, this diagram, just to represent the different veins. Here the first, at the dorsal part, the C, the costa. Then CS represents the subcosta, R radius, and the radial sectors represented by here RS. In this diagram, the media is represented by the M, cubitus by CU, anal by A, and their branchings have been represented in this diagram. And the status of the vein as a convex and the concave is also being represented in this diagram in figure B. But besides this basic organization, some other cross veins have been also represented, which was not primarily given by the Comstock and Needham. These are the modifications, and so many entomologists have reported such type of modifications. After Comstock and Needham, the major contribution given by another entomologists, and they are the Lamecri, Hamiltons, Tilliard, Segui, Raggi, and Clausens, etc. But what is the reason of these modifications? The major reason of the modifications are addition and deletions. The differences indicates two different methods of specialization by which this primitive type has been modified. The one specialization by reduction and the another specialization by additions. A reduction in the number of wing vein takes place in two ways. First, by atrophy of the vein, and second, by coalescence of two or more adjacent veins, meaning merger of two or more adjacent veins. The first method is illustrated in most of the orders where a reduction in the number of wing vein has taken place by atrophy, more or less complete, of one or more of the anal veins. This is correlated with the reduction in the extent of the anal area. This method is also illustrated in certain cases where there is no apparent reduction of the area of the wing from which the vein has disappeared. The most familiar illustrations of this occur in the Lepidoptera. The second method is the reduction. In reduction by coalescence, some veins fuse together And the process takes place in all of the orders in which the number of wing vein is less than in the typical wings. This also takes place in two ways. First, the point at which two veins separate occurs nearer and nearer of the margin of the wings until finally when the margin is reached. A single vein remains where there were two before. Second, the tip of two veins may approach each other on the margin of the wing until they unite, and then the coalescence proceeds towards the base of the wing. This is commonly found in the case of the dip. And in order to provide the rigidity, the cross veins between veins have been also reported. Here the cross vein, so many cross veins have been reported by several entomologists. The first one is the humeral cross vein. This is a single cross vein extending from the subcosta to the costa near the humeral angle of the wing. This is the most constant of all the cross veins. Then, second was the radiomedial cross vein represented by S. This is a cross vein extending from radius to media, usually near the center of the wing, and is designated by the RM, small rm. Third one is the mediocubital cross vein. Here this is cross vein extending from media to cubitus, usually near the center of the wing. It is designated by the abbreviation MCU. When in its typical position, this cross vein extends from a point near the base of M3, M4 to a point near the base of CU1. The next is the medial cross vein. The medial cross vein extending from the medio 2, M2 to media 3, M3. This is designated by the abbreviation small m. The presence or absence of this cross vein is often a character of the considerable taxonomic importance. Besides this, the auriculus have been also reported by some entomologists in many insecta. There is a cross vein extending from radius to cubitus near the base of the wing, and this has been termed as auriculus. Here, in some insects, jugal lobe of wing is occupied by network of irregular vein, but sometimes two distinct small veins have been observed. That is, first vena arcuata and the second vena cardinalis. But another important point here, some modifications have been reported besides this basic structure. And before considering the modifications, some furrows have been reported. What are the furrows? There are several kinds of the furrows have been reported in this system. The first one is the anal furrow. This is longitudinal furrow which is usually between the cubitus and the first anal vein. Second one is the median furrow. This is longitudinal furrow which is usually between radius and media. It is well marked in many of the hemiptera, where it is separated the embolium from the remainders of the corium, and in the hymenoptera its course is marked by a series of weak spots in certain vein. Third is the nodal furrow. Here this is a transverse suture beginning at a point of the costal margin of the wing corresponding to the nodus of the odonata and extending towards the inner margin of the wing. It crosses a varying number of veins in different order of the insecta. So several furrows have been also reported in the cross veins. The furrows of wings are in no sense homologous or even analogous to the veins. But the variations here we are going to consider in different groups of the insecta, like first in the Orthoptera. in the Orthoptera, how it differs from the basic concepts of the construct and Nidham, and probably this is a result of the evolutionary process. The major variations as a principle and increase in the venations have occurred by the branching of existing Comstock and Nidham vein. The branching resulted in development of accessory veins. Additional intercalary veins have also evolved in hindwing between definite veins. An anterior media absent due to reduction, but single posterior media existed. Vena dividius, a secondary vein, evolved in the hindwing, and the anal vein in anal lobes have increased greatly. So these are major modifications of the Orthoptera. This diagram represents all the modifications showing the wing of the Locusta. Locusta is the member of the Orthoptera with venations here. Some accessory veins have been represented here like the ACC, and every information we cannot consider here to represent in this diagram because lot of branching and lot of degenerations in the venation system have been reported in the insecta. Now in the case of Diptera, in Diptera again, some major modifications are: the first, the general atrophy of the cubitus and vestigial nature of 2A, 2 anal and 3rd anal are important. In Nematocera, R1, meaning the radius 1, is 3 branched. R2 and R3 are distinct. In Brachycera, R3 having 3 branched, but R4 and R5 are free. Similarly, in Anisopus, the single vein R2 and R3, due to merger of the R2 and R3, again the R4 and R5 have been merged together, and reduction in the anal vein also have been observed. In Tabanus, apical fusion of CU1, cubitus 1, and first anal resulted CU1 plus 1A as a single vein at the apical fusion point. R2 and R3, the radius 2 and radius 3, fused to form a single vein, and reduction in anal vein have been also observed. So in Diptera again, lot of variations have been reported. This is the basic diagram showing the venation system in the Anisopus, and this diagram showing the variations from the basic Comstock and Nidham arrangements. And this diagram showing the variations from the basic Comstock and Nidham arrangements in the case of the Tabanus. Now in the Plecoptera. In Plecoptera, in the most genda, the anal area of the hindwing has been extended and the number of the veins increased. Here, in the nymph of Pneumora, the costal trachea disappeared, the subcostal is forked, and vein SC2 anastomosis with the vein R1. The radius is reduced, and the radial sector being only 2 branched, the media is reduced. The cubitus is typical, but in forewing several cross veins have been developed between its branches and also between it and media. In hindwing, the 2nd and 3rd anal veins are forked. So especially in the order Plecoptera or stonefly, in most genda, the anal area of the hindwings have been expanded and the number of anal veins increased. In the pre-anal area of both wings, the number of wing veins has been increased in certain genera and reduced in others. This fact, taken in connection with the generalized condition of the basal attachment of the trachea of the wing, already pointed out, leads us to believe that the Plecoptera as a whole depart less widely from the primitive winged insect than do the living representatives of any other orders. Now, in the Hemiptera, in the case of Hemiptera, The wing of young nymph and trachea that precede the primary veins are most massed, but in older nymph, the cross veins contains no trachea. In wing of the mature nymph, the costal trachea extends nearly to the apex of the wing. The subtrachea is also prominent but not forked. The radius is reduced to three-branched condition. The first anal trachea coalesces with the cubital trachea, and the second and third anal trachea are also united at the base. So in Hemiptera also, there is a lot of modifications, and a study of the wings of Hemiptera reveals remarkable departure from the primitive type of the wing venations. Now, in the Heteroptera also, lot of modifications have been reported. Like in Pentatomid nymph, the costal trachea is well preserved, but subcostal and radial trachea are closely approximated. In distal half of the wing, the subcostal traverses that part of the wing which should be traversed by the trachea R1. And the trachea that precedes the radial sector is two-branched. The medial trachea is six-branched. And in the hind wing, the radius has been highly reduced. So in Pentatomid nymph, it is evident that the supplementing of R1 by the subcostal takes place here. And such type of supplementation has been widely reported in the case of the cicads. The trachea that precedes the radial sector has its characteristics bent at the base and is two-branched. Now, in Lepidoptera, particularly in the Astenopeus, in the forewing vein M4 and Cu1 coalesces, merges together for the greater part of their length, and one of the anal vein has been lost. In the hindwing veins M4 and C1 anastomoses, but separate near the margin of the wing. In Peronoxystus, the R3 and R4 are anastomosed. The base of the vein M2 and M3 have migrated towards subcutus. In the hindwing again, there is modifications. In the hindwing, a great reduction in the subcosta radial area of the wing has taken place. First vein, subcosta and radius merges together to form the margin of the wing near to the base of R1, and second, the radius sector is reduced to single vein as RS. Now, conclusive remark. Here the major conclusion is, still there is a lot of research is needed. The basic concept given by the Comstock and Needham is still running, but evolutionary process is going on day by day in the case of the insecta, because insects are very susceptible to the natural selection process and very frequently modifying its morphological structure or anatomical organizations also. So definitely in the course of the evolutionary process, a lot of changes in the venation systems occur. So there is a need of further study regarding the venation system of the insecta. Normally the faculty members and the postgraduate students are collecting different insects for the study of the biodiversity, especially the Lepidopterans and Coleopterans and other insects also. Definitely we should give some attention towards further modification, towards the recording of further modification in the venation system of the insect, and for this study very advanced laboratory is not required, only simple microscope is required. So we should definitely contribute in this field, because this field is again having very large scopes of contribution. J.H. Comstock and J.G. Needham has contributed in 1898 and still we are totally dependent upon this hypothetical system given by the Comstock and Needham. So definitely there is a wide barren field for further research study. Finally, thank you. I hope the information will be contributory for your study. Thanks.